Hello everyone. In this lecture we are going to discuss about disaccharides. So in the previous lectures we have seen about introduction to carbohydrates and classification of carbohydrates, about monosaccharides, classification of monosaccharides, examples and their structures. So this lecture is a continuation for the previous lectures. So disaccharides are the carbohydrates or the sugars which consists of two sugar units in their composition, which means when you hydrolyze these disaccharides it will give monosaccharides. So di stands for two, right? So di stands for two and the saccharides stands for the sugars. So disaccharides are the carbohydrates which consists of two sugar units in the structure. Disaccharides are of two types. One is non-reducing disaccharides and reducing disaccharides. The non-reducing disaccharides have the C1 glycosidic linkage, also called glycosides having the C1 glycosidic linkage, which means the glycosidic bond forms between the two sugar units. At one sugar unit, the glycosidic bond will be formed between carbon atom 1 of one sugar unit and carbon atom 1 of another sugar unit. So the example of non-reducing disaccharides are the sucrose. So another one is reducing disaccharides, also called glycosyl aldoses or glycosyl ketosis. So again these are of two types. One is C1 glycosidic linkage disaccharides, which means lactose, maltose, cellobiose. So this disaccharides consists of C1 glycosidic linkage in this structure, which means the glycosidic bond forms between the C1 carbon atom and C4 carbon atom of two sugar units. Similarly, C1C6 glycosidic linkage is present. Examples are genteobiose and melobiose. So the glycosidic bond forms between the C1 and C6 carbon atom of two sugar units. So when you see the example, so what are glycosyl and glycosides? So often simply referred to as diglycosides or oligosaccharides. So these are the complex carbohydrates where a sugar molecule, which is the glycosyl group, is linked to another sugar unit called the glycoside via a glycosidic bond. So basically these are act as a strong storage compounds in plants, animals and microorganisms, often improving their structural diversity. So let's see some examples of C1 glycosidic linkage. So this is the structure of a disaccharide having the linkage between alpha D glucose and beta D fructose. If you can observe in this structure, the glycosidic bond formation is present between C1 and C1 glycosidic linkage. If it in case of C1 and C4 glycosidic linkage, you can see here, this is the carbon atom 1 and this is the carbon atom 4 having the glycosidic bond between them. So this kind of linkages can be observed in case of reducing disaccharide, and C1 glycosidic linkages can be observed in case of non-reducing disaccharides. So let's discuss an example. So sucrose is a non-reducing disaccharide. Basically it is a disaccharide commonly called a cane sugar. So as sucrose consists of two subunits in its composition, upon hydrolysis it will give alpha D glucose and beta D fructose in equal amounts. It is a common sugar. It is widely distributed in photosynthetic plants, and this is the only foodstuff used in crystalline form. It is the predominant form in which the sugar is transported from leaves to other organs of the plant through the vascular system. So sucrose actually is the most abundant form, right? So this most abundant and widely distributed form in photosynthetic plant has to be distributed or transported from leaves, which are the food factories of the plant, to the other organs of the plant through vascular systems. So what is vascular system? The vascular system consists of vascular bundles. So the vascular bundles are formed by the tissues known as xylem and phloem. So this together forms the vascular bundles or vascular system. Through this vascular system, the sucrose is transported to the other parts of the plant. So sucrose on hydrolysis by dilute acids or the enzymes like invertase, also known as sucrase, gives invert sugars. So sucrose is the mixture of glucose and fructose. So the mixture of glucose and fructose gives sucrose. So the sucrose is a mixture of glucose and fructose. So the glucose is dextrorotatory, which means it can rotate the plane polarizal light towards the right direction. That's why it is known as dextrorotatory. And fructose is levorotatory, which means it can rotate the plane polarizal light towards the left direction. So as these are the two different sugars which rotates the plane polarizal light towards two different directions, so the sucrose gives invert sugars upon hydrolysis. So there is an inversion of sign of rotation, as I have mentioned. So the mixture is called an invert sugar. That's why the sucrose is known as an invert sugar, gives two invert sugars upon hydrolysis. So it is sweeter than lactose, maltose and glucose. It does not possess mutarotation. So it does not possess a mutarotation. Why? Because it don't have an anomeric carbon, and it is not a reducing sugar, and it is not at all reactive. So why the sucrose won't be able to exhibit the mutarotation? Why? Because it don't have the anomeric carbon, and it don't have a functional group like aldehydes and ketones. That's why it won't be able to react with other compounds to form the other products or isomers. If you see the formation of sucrose, it is formed by the elimination of a molecule of water from glycosidic hydroxyl group of alpha D glucose and beta D fructose. So if you see this reaction, sucrose is a mixture of alpha D glucose and fructose, right? So in order to form the sucrose, a glycosidic bond or linkage must be formed between this glucose and fructose. So the linkage is always C1 and C1. You can see here how the glycosidic bond will be formed between these two units. It is dehydration actually, which means the loss of one water molecule. So these are the hydroxyl group present here. So in hydroxyl groups will be come out as a water molecule. So H2O will be lost like this, and finally a glycosidic bond will be formed between these two sugar units at C1 and C1. Finally, upon the loss of water molecule, there will be the formation of glycosidic bond here at C1 and C1. This is the carbon atom 1 and it will be the 2. So don't confuse, this is the carbon atom 1, the linkage is like this. So this is the glycosidic linkage which forms a sucrose. So sucrose is the non-reducing disaccharide. So this is a brief introduction about the disaccharide and classification of disaccharide and about non-reducing disaccharide sucrose. I hope you understand this lecture. In the next lecture, we will continue about the reducing disaccharide units. Thank you.